For this presentation, we're going to be learning how genes are passed from parents to offspring. Every code that provides you with your traits is in your genes. For example, brown eyes are among the many options for eye color. You receive one half the code from mom and one half from dad. Depending on how these codes match up, you may have brown, blue, green, or some other variation of eye color. Let's take this concept and apply it to Gregor Mendel and his famous pea experiments. In 1854, Gregor Mendel researched how traits were inherited by plants. He was a monk and did his research at his monastery. At the time, it was believed that offspring would inherit a blending of the traits of each parent. Over 8 years, Mendel studied inheritance by working with pea plants because they were easy to breed and they had a variety of traits. In his experiments, Mendel found that the traits for pea plants could be dominant or recessive. Some of the examples of the traits that he looked at was the plant trait for height. Tall plants were dominant to short plants. He also looked at colors, where purple flowers were dominant to white flowers, and green peas were dominant to yellow peas. There are many more traits that pea plants exhibit. Based on his finding, Mendel developed the following laws. The law of segregation, where two parts of a gene pair or allele separate from each other in the formation of sex cells. Half the sex cells carry one allele and the other half carry the other allele. The law of segregation occurs in the first stage of meiosis, and that would be meiosis 1, specifically in mitosis 1. The law of independent assortment states that traits are passed on independently of other traits from parent to offspring. Again, this is in meiosis 1, when the tetrads or the homologous chromosomes line up on the metaphase plate. They don't necessarily have to line up in any specific order. They can line up randomly, independently of the other chromosomes. In Mendel's experiments, when he found that certain traits were dominant or recessive, this means that the dominant trait is the one that showed up in the population more often, while the recessive trait was hidden or receded into the background. We call those dominant and recessive genes alleles. Depending on how those alleles pair up, you may see the dominant or the recessive trait. This next code is followed throughout all genetics. The dominant alleles are represented with a capital letter always, and the recessive alleles are always represented with a lowercase letter. For example, we can use capital B and lowercase b. Two of the same alleles are considered homozygous or purebred. That would be a big B, or a little b. Two different alleles are considered heterozygous or a hybrid, and that would be big B, little b. Putting them together can make several different combinations. When we have a homozygous dominant, we have two dominants, and we would see the dominant trait. A heterozygous genotype is where the dominant is going to cover up the recessive allele, and we would still see the dominant trait. A homozygous recessive with two recessive alleles would show the recessive trait. When we put it all together, we look at the letter combination for the trait of height in pea plants. The letter combination of alleles is called the genotype. For example, in the tall versus short plant above, if the big T is dominant and the little t is recessive, what would the genotypes be? For the dominant tall height, it would be big T, or big T, little t, as the big T overshadows the little t. And in recessive, it would be little t for the short plant. The physical expression is called the phenotype. For example, the phenotype is what we physically see with our eyes. So in the pea plant example above, what would we see for the dominant trait? Tall, and the recessive trait would be short. Now we're going to practice as we learn about young rat love and apply this to Punnett squares that you learned about back in middle school. We're going to do this example together. Two young rats are in love and want to start a family. Let's see if we can predict what their children will look like. We'll start by looking at the alleles that control fur color. Keep in mind that a rat has two genes for every trait, one from mom and one from dad, and one of those two genes gets passed along to its offspring. We have a male with the genotype big A, little a, which is the agouti a brown and black mix phenotype, and the female has a genotype a, and has a black phenotype. Let's figure out what fur color their offspring might have. To do this, we are going to use a Punnett square. You have a Punnett square on your page, and you're going to use this as we go in and fill out the different boxes. Punnett squares are useful genetic tools in helping us predict the possibilities of the offspring genotype combinations and phenotypes. So you're going to start with a blank box on your page. We'll put the male's alleles for big A, little a at the top, one allele above each box. As the female's alleles, little a, on the left side, one beside each box. One allele per box, because remember the alleles separate during the law of segregation in meiosis. Now you're going to carry the big A of the male allele into each box below, and carry the little a allele of the male into each box below. Once you finish the male, do the female. Carry the little a allele at the top to the next two boxes over to the right, and the bottom little a, the next two boxes over to the right. And this is going to fill in the different combinations that we have for our offspring. Next, we're going to see what the actual phenotype would be since we have the genotypes. We have 50% big A, little a, and 50% little a, little a. This shows all the possibilities available for this pair of rats. According to the Punnett square, 2 out of the 4 boxes, or 50% of the baby rats, would have the genotype big A, little a, which is the agouti phenotype. Remember that the dominant trait covers up the recessive trait when they are together. And 2 out of 4 boxes, or 50%, would have the genotype little a, which is the black phenotype. So now we're going to practice. Identify whether each is homozygous or heterozygous. Do this on your own, pausing the video, and then when you are ready, hit play. You want to check and see if you got them right. The HO stands for homozygous and HE stands for heterozygous. Okay, here's another practice with a Punnett square. You want to find the probability of offspring for each problem below. You are mixing a male dad who is big D, little d, with a female who is homozygous recessive. Think what homozygous means and what recessive means. Put the two together, and that tells you the female is little d, little d. We want to figure out what the female's genotype would be. So again, homozygous recessive tells you little d, little d. You want to draw in your own Punnett square. Separate the male's alleles across the top and the female's alleles across the side. Once you finish this, you're going to fill in the Punnett square. Check to see with Miss Frunheiser if you're correct.